All right, so welcome to the second segment on the supergroups, but the third segment overall on protists. So we're going to be discussing the SAR clade of protists. We can see that the SAR group, again, as I mentioned in the first segment, includes the straminophiles, the alveolates, and the rosarians. So SAR stands for straminophiles, alveolates, and rosarians. The straminophiles are divided up into three categories based on this phylogeny, and that includes the diatoms, the golden algae, and the brown algae. And the alveolates include the dinoflagellates, the apicomplexans, and the ciliates. And the rosarians include the radiolarians, the foraminifera or forams, and the cercozoans. So here, this is the same slide as previously. We can see the list of supergroups in which the protists divide into, and I kind of explained the SAR clade. So remember, the four supergroups include the excavata, which were discussed in another segment, the SAR clade, which is for the current segment, and then these are the other two segments, which include the Archaeplastida and the Uniconta. So, as I did previously, I'm going to go ahead and take the SAR clade and show its relationship to the other three clades, and then divide it into a concept map. Okay, so we're going to continue with the protists again. Remember that we are dealing with the SAR group this time. So let's go ahead and use this color. Okay, so again, we'll start with the ancestor. Okay, eukaryotic. Okay, and then remember there are four. I'll just kind of draw this different, right? So you might want to do it differently. It depends on what your preferences are. Okay, so they all come from the same ancestor. We already covered the excavates. Now we're dealing with the SAR clade. And then remember we have the HAE plastida. And then we have the Uniconta. Okay, so the SAR clade divide into three groups. The straminophiles, straminophiles, and then we have the alveolates. And each of these divide into different categories, right? So we're going to further divide these in the rosarians. Okay, then the straminophiles divide into three. So this starts getting a little bit more complex, right? So we have the diatoms. And then we have the golden algae. And then we have the brown algae. And then the alveolates divide into three. And that includes the dinoflagellates, the apicomplexans, and the ciliates. And the rosarians are going to divide into the forams or the foraminifera. And the cercozoans. And then, of course, the radiolarians. Okay, so these are ciliates right here. Okay, so again, we have the SAR clade. Okay, so this is a monophyletic group. It's called a clade. And then we have, that's divided into straminophiles, alveolates, and rosarians. And you can see that SAR stands for the first name, so that might help you remember those. And then the straminophiles further divide into the diatoms, the golden algae, and the brown algae. The alveolates divide into the dinoflagellates, the apicomplexans, and the ciliates. And the rosarians divide As mentioned, the SAR clade includes the straminophiles, the alveolates, and the rosarians. It's quite a diverse group and a very large group. It is a monophyletic group still. However, the next two groups will include polyphyletic groups. Now, this group is actually pretty controversial regarding the evolutionary relationships, and their exact relationships, or many of their exact relationships, hasn't really been established yet. But let's go ahead and look at the characteristics of each one. So let's start with the straminophiles. Straminophiles include those organisms that many of them are photosynthetic, and they're really important photosynthetic organisms. Most of them have what's called a hairy flagellum, because I mentioned that previously when we were looking at other organisms, but they have a hairy flagellum paired with a smooth flagellum. So you can see the two flagella here, one of them is hairy, one of them is smooth. The straminophiles include the diatoms, the golden algae, and the brown algae. Now, the diatoms are unicellular algae. They contain a glass-like wall made out of silicon dioxide. They're actually a major component of phytoplankton, and they're very diverse. You can see the diverse shapes, and this shows a scanning electron micrograph of one type of diatom. Again, they have many, many different shapes. Diatoms are important producers. After a diatom bloom, they start to die, right? And then they're going to fall to the ocean floor. And they decompose relatively slowly, especially in the ocean where it's salty water. Now, during their lifetime, remember, they're photosynthetic, so they take in carbon dioxide from the atmosphere, which is a good contributor to the production of oxygen in the atmosphere since they photosynthesize, and the reduction of carbon dioxide in the atmosphere. So they incorporate this into their biomass, right? And then it will be sequestered on the ocean floor for many, many years up to centuries. Now, some scientists believe it's important to actually fertilize the ocean floor with iron to promote these diatom blooms. Once the blooms start to die, then this actually facilitates the the movement of the carbon dioxide from the atmosphere to the ocean floor once they fall down to the ocean floor after they die. So this now golden algae, they're named, of course, for their color. You can see some examples here. They're yellow colored. This color is the result of their production of yellow and brown carotenoids. So these are pigments. Now, the golden algae are typically biflagellated, meaning that they have two flagella with both flagella on one end. Now, all golden algae are photosynthetic. Some of them are mixotrophic. So remember, they can combine photosynthetic or photoautotrophy with heterotrophy. Most of them are unicellular. However, some of them are colonial. So here you can see some colonial examples. So they just kind of live in colonies. Again, each of these are individual cells, but they're living together in a colony. The brown algae are the ones that have plant-like structures. So they have three structures that are analogous to plant structures. So the holdfast, which actually anchors the algae to some sort of substrate, like a rock or the bottom of the ocean. So this would serve like a root. There's the stipes, which provides support. So that's basically like a stem, right? So it allows it to kind of stand up. And then the blades, which are like leaves. Now, brown algae are not like plants. They do not contain true tissues and organs like plants do. Some of them have gas-filled floats, which are kind of bubble shaped, to keep their photosynthetic structures near the surface of the water to be able to obtain more light. Now, if we look at this structure right here, we can see that we have the holdfast, which again serves like a root, and it's anchoring it to the ocean, the bed, right, which actually contains some mussels and other animals. And then we have the stipe, which is like a stem, and then the blades, which are like leaves. So it looks like a plant, however, it is not a plant. Now, the brown algae have an alternation of generations, so we kind of discussed that a little bit. Remember, the diploid generation is the sporophyte, and that has 2 copies of each chromosome, and the haploid generation is called the gametophyte and has 1 copy of each chromosome. The sporophyte produces the spores, and the spores here are called zoospores. The spores are going to develop into the haploid male and or female gametophytes, which are going to then produce the gametes. So the gametophytes produce the gametes. Fertilization of the gametes results in a diploid zygote, which then grows into the sporophyte and then continues the life cycle again. Look at this structure right here. We kind of saw that earlier. We have the sporophytes and we have the gametophytes. And the sporophyte's going to produce the spores. And the spores are diploid, but then they're going to undergo, or the sporocytes are diploid, and they're going to undergo meiosis to produce the spores. And then the spores will be released. And the spores are called zoospores. Those are going to be released, and then they're going to land somewhere and germinate. So then they're going to either produce a female or a male gametophyte, which will then produce either the egg or the sperm. So we can see the egg and the female gametophyte, and then the sperm have flagella, so they're going to swim and find. And then once fertilization occurs, the egg develops into a zygote, and then further develops into a mature sporophyte. Okay, so the next major group of subgroup within the SAR clade is the alveolates. That's the A in SAR. They're alveolates because they have what's called alveoli. Alveoli are membrane enclosed sacs just present underneath, just under the plasma membrane. Now the alveolates include the dinoflagellates, the apicomplexans, and the ciliates. The dinoflagellates have 2 flagella, dinoflagellates. They have 2 flagella, and each cell is reinforced by cellulose plates. Okay, so their cell is reinforced by cellulose plates. The 2 flagella are often embedded in a groove that is present in the spherical cell. Now, they are abundant in phytoplankton, and they're quite diverse in terms of their lifestyles. So they are generally aquatic, but they could be phototrophic, mixotrophic, and heterotrophic. These are the organisms that cause the red tide. So it's not a bacteria that's in the sea when we have red tide. It's actually a protozoan, and it's dinoflagellate. Now, the apicomplexans are generally parasites of animals, and some of them can cause diseases in humans that are quite serious. They generally spread to their host as special infectious type cells called sporozoites. One end of the sporozoite is where the apex is. So this is this apex, and it has a complex of organelles and proteins that are specialized to allow them to penetrate the host cell and tissue. And that's why they're called apicomplexans, apical complex. So they have this complex at the apex. Now, the life cycles are quite complex, and it requires 2 or more different hosts. So an example, or a key example, would be the Plasmodium falciparum, which is the organism that causes malaria. Now, it requires mosquitoes and humans to complete the life cycle. So let's go ahead and look at. So here we've got inside the human, after the mosquito bites the human, this kind of completes it right here. We have, they're going to go into the liver, and within the liver cells, they're going to kind of develop into the merocyte, and then they're going to spread to the blood cells and get inside the blood cells, where there's going to be several different stages of their life cycle. Ultimately, they're going to develop into what's called a gametocyte, which kind of results from the bursting of the red blood cells. Gametocytes are going to further develop, and then they're going to undergo fertilization between the male and female type, and this develops in, and the zygote undergoes meiosis to produce oocysts, and basically develops into an oocyst, and then of course the sporozoites are going to be haploid, and then this completes the life cycle right here. So this occurs in the mosquito, and then this occurs in the human. The next group of alveolates is the ciliates. These are quite diverse, and they include organisms that you probably are familiar with. So, of course, they're named because of the way they move, right? So the structures they use to move are the cilia. So most ciliates are actually predators of bacteria and other protists. One key example is the paramecium. The paramecium is one that you've probably looked at ever since you were in elementary school. So the paramecium, or the ciliates, also altogether, they have two types of nuclei. They have the micronuclei and the larger macronuclei. And you can see they have plenty of organelles. And remember, the ciliates live in fresh water, so they could take on too much water. However, they have this special adaptation. It's called the contractile vacuole. You probably remember from General Biology 1. It's going to fill up with water, and then it basically contracts so that the water is expelled out of the cell to keep the cell from bursting in a hypotonic solution, which is. Now, let's look at the reproduction of the ciliate, specifically the paramecium. It undergoes conjugation and fission. So here, let's talk about this. Conjugation is a sexual process. So this is separate from, I guess, reproduction by fission. So let's look at conjugation first, and we'll talk about fission. Fission's pretty simple. So here we have two genetically different paramecium, and assuming that they're compatible, they're going to kind of match up, and then basically the micronuclei inside them are going to undergo meiosis to produce four micronuclei. Well, three of them are going to die, and then the one that remains, or degrade, then the one that remained is going to be copied, and then they're going to exchange between themselves one of the micronuclei. And then here we have the final product. So this final one is going to be genetically distinct from the initial two. So it's basically a combination in terms of mixing up the micronuclei. And then this organism itself, or any one of these first two, or the product of the conjugation, could undergo fission. So basically, what that involves is, again, the micronucleus, and that's going to just basically copy, and then they're going to copy enough of the micronuclei and the macronuclei in order to reproduce, basically double. And again, these are genetically similar to each other. Okay, so they're the same, and then what results after conjugation is going to be something that's genetically distinct from the original two. Now, Rosarians is the last group of the SAR clade. Many of them are actually amoebas, but they're not amoebozoans. So we have amoeba or amoeboid-like type organisms that are not actually amoebozoans. Amoebas are those types of organisms that actually have pseudopodia, and that's how they move and feed. So they're basically just like extensions of the surface of the cell. And pseudopodia means like fake feet. So that's, again, how they move around. Now, rosarians, they have, some of them, the rosarians have thread-like pseudopodia, and some of them have lobe-like pseudopodia. So the rosarians include three subgroups, and this is the radiolarians, the foraminifera or forams, and the cercozoans. So let's take a look at those. The radiolarians, these are the ones that have the thread-like pseudopodia, so they're not lobe-like pseudopodia. They have internal skeletons that are made out of silica. Okay, so these are some key characteristics of the radiolarians. Now, the pseudopodia are actually reinforced by microtubules that radiate from the central part of the body. Forams, or foraminifera, are known for their special characteristics. They're basically they're shells, right? So they look like shells. They have porous shells in which pseudopodia extend out of the pores of the shells. The shells are called tests. The pseudopodia, of course, allow them to swim and feed, but also contribute to the formation of the tests. Now, some of them are going to be nourished from algae that are symbiotic with them. There are both freshwater and marine forms of foraminifera. Now, forams can form sediments that have been found in fossil record extensively. Now, there's magnesium content in them. It was used to actually has been used to determine the temperature of the ocean over time. Now, the cercozoans are a very large type of the rosarians, which are amoeboid in shape. So they do have like in terms of their body is amoeboid. Now, they do have a thread-like pseudopodia. Then they live in diverse types of environments like marine or aquatic, meaning marine and fresh, water, and also in terrestrial habitats like soil. Now, most of them are heterotrophic, and some of them are parasitic, and some of them are actually predators. So, here's an example of one that actually has a photosynthetic structure. It's quite unique. It's called a chromatophore. It's actually thought to have evolved from a different cyanobacterium than the chloroplast. Now, this is a summary of this SAR clade. Here we have, this is basically the major groups, and then there's subgroups, and then, of course, some major characteristics of the major groups, but not the individual subgroups. So this is where you want to kind of fill out that table a little bit more in detail and perhaps create a concept map to help remember the groups and the subgroups.